Hello, everyone. Welcome to the first Bet on Black pitch of the new year, 2022. We're so excited. And actually, I have some awesome news to share. Bet on Black pitch was officially trademarked. I believe Cherie may have shared it on a live yesterday, but it is official. 10K Project owns the trademarking rights to the term Bet on Black pitch. And so we are super excited. So now we can scream it from the rooftops. It is ours. And so if you hear anybody else screaming it from the rooftops, let your girl know, because they shouldn't be doing that. But so we're super excited. You know, it's official. We've been doing these pitches since last March. They were such a big hit in the last year. We really made an impact on many founders. And so we plan to do the same thing this year, but even bigger, right? We want to continue to grow membership so that we can continue to support the founders that are needing the help from us from a capital building standpoint. So with that said, I am going to get started. Tonight's founder is John Adamson, and John's company or the project name is DeoBioSciences. So I'm going to share my screen. And I'm going to show you the campaign page. You guys, as you know, we're going to put this campaign link in the chat throughout the evening, right? So you don't have to worry about that. It is a custom link. So John will be able to report back to you how many eyes we've actually sent to the campaign via this link. And so, you know, please copy that link that's in the chat if you want to go to the campaign page. So this is on Net Capital, and as you can see, it's $100 minimum. The deadline is January 26th. So we'll talk to John to talk about if there's any extensions or anything that'll happen, but the deadline is in 22 days, right? The share price is $50. John has already raised $83,450. You see the company's valuation. And then down at the bottom is where you'll see, and obviously you're going to read all of this, right? You're not going to just scroll to the bottom like I'm doing, but I want to show you where the Form C and all of the financial information is. So down at the bottom, you kind of get into where the numbers are. And then this is where you get into all of the legal documents. So here's the CPA review letter. Here's the Form C and any amendments to that Form C that John has. And so please take the time to read all of this information, right? To help you do your due diligence. It's important that you do so. As you know, here at 10K, we're not financial advisors. We don't endorse, but what we do is we bring great talent in front of you and investment opportunities that you then get to decide individually if you want to add that business to your portfolio. And so today we are highlighting DeoBioSciences. Super excited to turn this over to John to tell us about the amazing work that he and his team are doing in an industry that, as we all know, affects all of humanity. And so this is the first time we've introduced this industry to the 10K community, but super excited. So with that said, John, I'm going to turn it over to you, sir. I'm going to stop sharing so that then you can share, and I'll turn it over to you.
Okay, fantastic. Well, before I launch into the presentation, I'm just going to quickly say that thank you very much, Tawana. I really appreciate the introduction, and also you're allowing me to address your audience. Really admire what you do. I admire what the 10K is about. I think it's long overdue in American society that something like this existed. Crowdfunding, equity crowdfunding hasn't been around those many years, but it's still been around long enough, and or at least the opportunity, the concept of investing, and in companies with growth potential in different forms has been around. And I think that this is leveraging what I and some other people who are far smarter than I am think is really a transformative opportunity for particularly, I say, African Americans to have potentially transformational, we were just talking about transformational generational wealth building. And we're talking about actually creating a legacy for descendants far down the line. And that's the type of, not to minimize or trivialize any other type of investment opportunity, but this has the kind of multiplier effect and potential that VCs look for. And that's why VCs are in this game, because they're not looking to just get a nice return or solid S&P type return. They're looking for, they want obscene wealth opportunity. And we frankly would be unwise, I think, not to at least consider the same. You know, what you do with the wealth, I think it's bad perhaps just to go for obscene, just strictly for obscene wealth opportunity if you're that superficial. But it's getting wealth is not, I think, is not a moral problem. It's what you do with that wealth that is the question mark. And I think there's nothing wrong with a good person accessing and generating a lot of wealth. So I'm proud. Thank you for what you're doing. I'm very honored to be part of it.
You're very welcome, sir.
Yes. Yeah, my name is John, and I co-founded DeoBioSciences years ago after my following my first real job in life at Lucid Technologies, Bell Labs in New Jersey. And DeoBioSciences is let me just give you a high level concept overview. It's a stealth mode virtual biotech company developing an exciting preclinical drug, cancer drug, with the goals of selectively killing multiple types of advanced and metastatic stage cancers in humans, and we hope and project dogs and cats as well. Companion animals is what the term is. And we are reverse engineering the drug from anecdotal data and reports of human efficacy and safety involving past members of a remote rural community, and also corroborating studies from respected laboratories involving molecular homologs and humans and animals that further confirm the safety and efficacy of the data that we originally investigated. And so far, lab results conducted and vetted at Cornell University have met all our predictions for safety and performance. So right now, the reason we're here is that we're looking for risk-taking change makers to share our vision and join us in trying to further de-risk this compound and improve commercial viability. So with that, I just wanted to give you a 50,000 foot, and let me see if I can find the share screen here. Okay, all right. And here we go, share. All right. Does it look like do you see what I see? All right, good. All right, so where cancer research matters. Let's see if we go to the. By the way, can you also see this cursor that's dancing around here? It's blue, sort of blue, teal color.
It's like a blue dot. Yes, we can see that.
Okay, great. So make sure it's not good. All right, here's the mandatory legal disclaimer language I put up in the front of any presentation. And put there for a second. You can all speed read. All right. And the fact that you've had it presented to you means you've been duly informed, and we'll just, it's basically forward-looking statement type stuff and disclaimer language. So we'll just move to the next one. Here's the agenda for what we want to cover in this presentation. 3 sections we want to address, 3 general concepts and phases. One is we'll examine the market that we believe is worthy of our investment. The problem from the human side, from the veterinary side, and what we propose as a solution in the form of our compound called DBX31. We're going to look at our progress to date. What have we achieved so far? And what have we determined about our molecule and why we believe it warrants, why it deserves our attention and investment. And then we're going to look at our next steps. What's the game plan, short-term game plan going forward? So, it's important that you have a market that matters, right? A market that needs you and that matters to the world. And that's certainly the case when it comes to cancer. First problem is human cancer. Many of us are familiar with these sort of gross high-level statistics, but it's still, despite all the hype that's out there about miracle drugs and breakthrough advancements, in 2020, cancer still afflicted 20 million per year globally, or approximately 20 million, and 1.6 million per year in the US. In that same time frame, it killed 10 million per year globally, and approximately 600,000 a year in the US, actually just over 600,000. The key point is that standard treatment options, including the immunotherapies that we see advertised by farmers on TV so often, are inadequate for treating in a reliable, consistent way advanced and metastatic stage cancer. So this is a problem that still is present. It's still significant. It's life-threatening. It globally will personally affect 2 out of every 5 people in their lifetimes. And it will indirectly affect virtually everyone else, either through their relationships with these individuals and or through with their direct relationships and or through the economic impact it has on all of us. The second problem is veterinary cancer. A lot of people aren't aware of this, but cancer is a major problem in dogs and cats. It has certain cancers, in fact, have an extremely high prevalence among those dogs and cats, those veterinary patient populations, at a much higher rate than it afflicts even human beings. Cancer is a leading killer of the 67 million pet dogs in the US. Also, 6 million new cancer diagnoses are made in dogs and cats, each one of them each year, not collectively, but in dogs, 6 million, in cats, 6 million. More treatment options are needed. Our proposed solution, DBX31. It's a unique, it's a, let me just move this out of my screen here, it's in the way here. All right, DBX31 is a unique naturally occurring biomolecule that exclusively attacks a wide range of cancers through targeted extrinsic apoptosis, also known as cell suicide. This is a superior method to, as compared to other anti-cancer agents. Here's an example, by the way, just an illustration of cancer cells that are covered by very unique receptors. And these receptors were part of a theory that we developed that might provide exploitable pathways for triggering receptor-mediated apoptosis, also known as programmed cell death, extrinsic apoptosis that I just referenced above. Let's just have a quick biology lesson, a refresher for those who already had this as part of their background. Apoptosis is a process of cell self-destruction that is highlighted by the fragmentation of nuclear DNA. That's DNA that's in the nucleus. It's also called programmed cell death or cell suicide. Apoptosis is a preferred way to kill cells. There are a couple of ways to kill them. Generally speaking, there's apoptosis and there's necrosis. And apoptosis is the far and away preferred way to kill cells. Within apoptosis, there are 2 categories types of apoptosis. Generally referred to as extrinsic apoptosis and extrinsic apoptosis. Think of it, it's just like it sounds: internal apoptosis, external apoptosis. Extrinsic apoptosis is hard to achieve, but it's regarded as the holy grail of cancer treatment because it's a vastly superior option for killing cells, particularly cancer cells, that currently doesn't exist therapeutically. Here's just kind of a high-level illustration of the mechanism of action, MOA, or at least the theorized MOA of how DBX31 works, based on our observations and our observed properties in the research that we've done. DBX31 basically is what's called a ligand. It's a molecule that is injected into the patient and will, and by the way, the mode of administration can be, we believe, will consist of potential multiple routes, but for right now, I'm just going to simplistically say it's injected in there, all right? But that's actually an advantage that we'll just possibly discuss later that DBX31 also could have in terms of its long-term clinical potential. But bottom line is, it's exposed to cells, it drifts by the cell. On the exterior of the cell, there are these appendages, sort of like the ones we saw in that last black and white electron micrograph. And DBX31 is chemically attracted to bind to these appendages as they encounter each other and go by. And then it causes what's essentially called a death-inducing signaling cascade, a DISC, which is basically fancy terminology for it sends a message into the cell through this thing called a death receptor that causes the cell to engage in apoptosis. The critical thing here is that its unique molecular structure catalyzes an apoptotic chain reaction throughout the cellular interior. And because of its unique structure, not just any molecule can cause that to happen. And that's why, again, it's extremely hard for this reaction between a death receptor that connected to the extrinsic apoptotic pathway to be triggered by an external molecule. Let's go research to date. Right. So let's actually look at our data. We evaluated this work not in our own labs, not internally within our own company, but using scientific talent that was resident at Cornell University, whose scientists and researchers there. This way you can have the confidence that it's not John Adamson saying this or someone that John Adamson paid and hired. It's someone who is reasonably objective third party that was paid to conduct the research, but would not produce false results in exchange just for conducting the research there, because frankly, they're not paid enough to do that, to risk that sort of professional liability, among other things. So they were just to conduct the evaluation and determine what data does our research study produce? What does it yield? And this was done as arm's length assessment done by them against these 8 very robust, hard, treatment resistant, hard to kill cancer types. All right, let me just go through them here basically real quickly. Oh, sorry, I actually jumped the gun here, apologize. They consisted of a wide range of tissue types and histologies, covering everything from gynecological cancers to lung cancer, the type of cancer that anyone could possibly contract, male, female, etc., and colorectal cancer. Again, type of cancers that are widely found throughout society, and of course, particularly our community. The important thing about this is that these were all advanced stage at a minimum cell lines. These were not primary stage tissues. These were not easy to kill, easy to treat. This was basically equivalent of a killer's row of cancer types. It consisted of, many of them were metastatic as well. So that basically means that they had advanced to the point where they broke free from the primary tumor and they now were circulating through the body and trying to find other organs that they could establish distant metastatic cancers and tumors. That basically they were at the point that this is the cancer spreading. That's what the common term and phrase is for. So many of these were metastatic. It consisted of triple negative breast cancer, ovarian cancer, colorectal cancer, like I said, lung cancer, skin cancer. But the common denominator is that they were all, again, not easy to kill, not easy to treat. So how do we do? The summary results are that we induced apoptosis in all of the cancer cell lines, 8 out of 8, 100%. The apoptotic index, that is the percentage of cells that were killed out of the percentage of the number, total number of cells that could have been treated, that were subject to treatment and could have been killed, ranged from 42 to over 90% that were killed after only 72 hours of incubation. And side note, 2 cell lines were after 72 hours over 95% dead. The other critical point is that no adverse effects occurred on normal non-pathogenic cells. High death rates for cancerous cells, no death rates for non-cancerous cells. Let's just drill down into several specific examples, just to give you a better flavor of what happened, what's significant about what we did here. This is advanced stage lung cancer, one of the tests we performed. There are 3 micrographs. These are basically photographs done through an electron microscope of fragmented cellular DNA and apoptotic bodies of advanced stage small cell lung cancer via a Tinel fluorescence assay. These were taken at different intervals, but they showed a progression of apoptosis from the top going down in the form of increased intracellular cytoplasmic content, represented as the white stuff that you see in the background. It increases as you go from top to bottom, representing the progression of advancement of apoptosis. Now, the thing that's impressive about this is that lung cancer, we all probably know, was it has historically been the highest, the highest represented, most prevalent, and the highest mortality type of lung cancer, both in the US and globally. That's recently been overtaken by breast cancer. But historically, lung cancer has been very lethal because it's very hard to treat. It tends to be very resistant to treatment, and usually the prognosis after a diagnosis, usually not very long, especially at advanced stage, is usually not many years. The type of lung cancer in particular that is reflected here is the toughest to treat of the teenager type of lung cancer. A lot of commercials you'll see that talk about lung cancer being treated on the related to a lot of the immunotherapies, talk about non-small cell lung cancer. That's easier to treat than small cell lung cancer. This was small cell lung cancer. And again, we obtained very high apoptotic kill rates on this assay, on this test. Here's another test, metastatic triple negative breast cancer. The thing about triple negative breast cancer is that it is the most lethal, the most treatment resistant, the most basically, well, it's the most difficult to manage breast cancer prognosis that you can get. The reason it's called triple negative is because it is missing. That means it's negative for the 3 major receptor targets that are normally attacked for any therapeutic regimen on breast cancer. Either estrogen receptor positive, progesterone receptor positive, or HER2 positive are the targets, or the normal receptor targets and treatment targets for the other types of breast cancer. Triple negative has none of these targets present, so that if it's in an advanced stage, there's no way to induce apoptosis in it or treat it because it usually is at the point that intrinsic apoptosis, because it's at an advanced stage, will no longer be affected. And the other targets that you would normally attack are not present on it. So again, of all the breast cancer types, it is the most lethal. And here's another important point for this audience. Triple negative breast cancer very, very disproportionately attacks or occurs in color, including African-American women and many women from subcontinental Asia. And it also tends to occur disproportionately in younger women. So it's a very important target for treating breast cancer. Here's what we did. You can see basically, again, just like the last one, this is a black and white electron micrograph. The white represents the amount of basically interior cell contents that's being exposed. This basically is a good thing. That means we're killing a cell. And you can see it starts next phase of it. In the final phase, it explodes, and basically it's considered to be just exploded apoptotic debris and nuclear and intracellular contents. It's cellular disintegration. Bottom line, it's dead. Here's 3 out of 8. How do we say, well, you might say, okay, you did a pretty good job against some of those other cellular, cancer cell lines, but how does DBX31 compare to promising experimental cancer drugs that are competing and that have shown efficacy against the same cell lines that you're addressing? And that's a great question. So here's an example of a drug called selenolysin. It's an antibiotic that was widely studied as a potential anti-cancer drug, and is still actually studied across many laboratories across the world. It's not owned by any particular company. Now it's an antibiotic, so I believe it's off patent, so it can be accessed and used for research by different labs. The bottom line is that it's widely studied and considered a promising drug candidate by a lot of different labs, different countries. Here's a study of out of Germany, and here's the citation for the study and results in case anybody wants to look it up themselves. But here's the graph of how the selenolysin, their drug candidate, performed against a cell line of advanced stage colon cancer, HT29. That happens to be the same colon cancer cell line that we assessed against DBX31. So here's a graph of the percentage of cell death that was caused by their drug candidate, HT20 SAL against HT29. In 24 hours, you can see it killed just under 40% of cells via primarily necrosis, not apoptosis, necrosis. DBX31 was tested against the same cell line. It killed over 90% within 72 hours via apoptosis, which is a vastly superior of killing a cell compared to necrosis. The difference between 24 hours versus 72 hours is something that I can't explain or account for because they only provided data for 24 hours. They didn't provide a 72-hour profile. My assumption is that they provided the best data they had. I think if they had much better data at 72, it makes sense that they would share it, but the chances are is that they pretty much plateaued after 24 hours and there was no upward trajectory or slope that would indicate higher kill rates at 72. So it's fairly safe to say, look at the data, that 40% is approximately where they topped out. So we don't think 72 hours would have made a significant difference, but still over 90% within 72 hours against that same cell line, especially when we did it, achieved it via apoptosis and not necrosis, is significant and dramatically superior outcome. Let's just finally cover our showcase experimental test. This was DBX31 versus SKOV3. SKLV3 is a cell line used as a negative control for the presence or absence of P53. This is fancy talk for explanation that it's virtually invincible to anti-cancer and pro-apoptotic drugs and toxins, including a extremely lethal cytotoxic cocktail of diphtheria toxin and ricin. That's actually used in the terrorist circles in the real world as a bioweapon. Actually, it's not, ricin is usually used itself. It's not usually combined with DTX. So the fact that you're combining these two makes it an extremely virulent and extremely dangerous compound. It's generally very, very good at killing cells. It's not intended to be used as a drug. This is not a cancer drug. No one is given DTX ricin for any therapy. It's dangerous. It is a pure bioweapon. But the example is simply being provided to show that even against something that is not designed for any therapeutic purpose, for cancer drug treatment, we want to see how DBX31 performed against compound, that agent. Again, let me just explain the point, reemphasize. DTX ricin SKLB3, this ovarian cancer cell line. And the upshot is that SKLB is a cell line that's highly mutated. It's so distorted. It has so many deviations in it that it's basically, it barely resembles a human cell anymore. So it's basically, if you will, almost like a Frankenstein monster of cancer cells. Here's how we did. DBX31 successfully caused robust apoptosis in these SKLB3 cells where all other cytotoxic agents, including DTX ricin, failed. We had apoptotic rates exceeding of SKLB3 cancer cells were dead after only 72 hours. And again, no adverse effects occurred on normal non-cancerous cells. Here's an example of here's the curve of the time lapse and how it changed over time. And even though we stopped assessing it at 72 hours, it was in an upward trajectory. So you can see it's an extremely high probability if we kept measuring it cycle, it's very high that would not have just dropped off. There was no indication at all that it was trailing off. It's very probable.
John, I think you froze. Okay, sorry guys. Let me let John know he froze. I wasn't sure if it was me or John. Let me get him back in. He may not realize he froze. Okay, I just sent him a text. That was like that was intense. That was an intense science lesson. Like, I feel like, but I was getting it, but I feel like, whoa, I was sitting here like, whoa, wow. And you guys already have some really great questions in the Q&A. So if you, as you're thinking of them, put them in there. So when we get to that Q&A time, he can ask, he can answer the questions. But yeah, it wasn't it? Yeah, it was. I thought I was the only one. I was like sitting, I took myself off camera and I was sitting here like, look at look how it dispersed everything. Like, wow, it's just, but he did a great job explaining everything and making it like very simplistic, right? Because we're all, well, some of you may be scientists. I know I'm not a scientist. He actually made me understand it. So hopefully we can get him to come back in. Saw my note. Let me try to call him. Let me try to call him, guys. Hold on one second. I do apologize for this delay.
Hello? Hello? Can you, Tawana,
Yes,
can you hear me?
we can. Yep, I was just trying to call you, John. We can hear you. I can't see you yet, but I can hear
Yeah.
you.
I'm gonna re-put it back on the, let's see, start my video. Apologies here. I've got Comcast. I'm not gonna necessarily blame them for everything.
It happens. It absolutely happens. But we were just saying how like the great information and how intense it was, and we were all like on the edge of our seats, listening and learning from you this evening. So I'm glad you were able to get back on.
Well, I am too. I've dropped that. I went to a mobile hotspot just not to take any more chances with it. So as long as it looks good right now.
It does indeed.
Okay, great.
So you want to share your screen again?
Yep. Okay. Let's see. Get here. You can see the launch meetings. Let's see if I'll get back to the, all right, here we go. All right, let's try to get back here. We're right here. I think we're at the conclusion.
We lost you. I think you were on.
Was it the graph?
Yes, you were still on the. Oh no, we lost you. You were still talking about SKOV, and you were showing us the bar graph, and you were talking about the fact that had you tested it beyond the 72 hours,
Okay, gotcha.
how it was on the upward trajectory.
Yep. Okay. Well, I'm right past. That
Okay.
was it. Let's go right past. All right. Great. Thank you.
See, I was listening.
You still were. This was all a test, Tawana, and you passed. Good job. A for you. It was a pop quiz. You got it. All right. All right. Let's get here. All right. All right. Move this to the side. All right. Let me just move here for a second. All right. Okay. So let's get past that to the next one. And the conclusion. All right. This is probably the part that we dropped out at. So against SKOV3, DBX31 outperformed two of the top selling chemotherapy drugs. ADM and CDDP, known as adriamycin, and CDDP, cisplatin, and a cytotoxin that's used as a bioweapon. That was the DTX-Ricin, without harming normal cells. This data was basically in a study, the efficacy of these other compounds against SKLV3 was confirmed in a study by UCLA Medical Center regarding an assessment of how SKLV3 is resistant to virtually all known anti-cancer therapeutics. There are certain parts of the cell line which can be susceptible to treatment by someone, but as a whole, unless you're selecting out those cells, unless you're just taking a cell, the cell line as it's found in its normal state, getting any kind of response from it is virtually impossible. But the point is, we outperformed this lineup of drugs consisting of both experimental drugs as well as FDA-approved chemotherapies. So what? What are our next steps? All right, well, it's all about de-risking. De-risking is the key. While DBX31 suggests amazing potential, we still need to de-risk, prove it with greater certainty, greater statistical certainty in particular. This data, the type of data that helps to increase statistical probability of future success, is the key to accelerated development and progress. It leads to more favorable opportunities for partnership, licensing, and financing. It makes hiring and expanding the future team with the best talent easier as well. So de-risking is the key. How do you do that? How do you de-risk without going all the way through clinical trials, which we all know be very expensive? Well, that's the beauty of where we are now technologically. Maybe 5 or 10 years ago, we couldn't say this, but today we actually have, we might not have the type of breakthroughs in cancer drugs that we want, but we do have enabling technologies surrounding cancer drug development that really have elevated what we can do by leaps and bounds. So whereas before, anything preclinical, you had to take with a grain of salt in terms of the quality of the data, in terms of its ability to predict what the drug is actually going to do at the final clinical phase, say phase 3 clinical stage. Today, there are clinical models, translational models, what they're known as, that can predict clinical success, final outcomes that the FDA will use to make a decision on approving a drug can be predicted with 90+% reliability at the preclinical stage. So essentially, your ability to make an investment in a drug, that is the risk that you would take by investing in a drug, can be dramatically reduced at an earlier stage of drug development. And that's what we were trying, that's what we're going to do at the next phase with using these highly predictive mice models. Patient-derived xenografts is the a term for an innovative platform technology that's able to accelerate the development of oncology drugs by using special human tumor xenografts in mice to predict their clinical effectiveness with 90 to 99% accuracy. So these models basically consist of taking extracting, usually through a biopsy or perhaps through a surgical procedure, and in the course of actual surgery, taking a piece of human tumor and implanting it into a mouse avatar, basically a mouse that you're going to use as a representation of a future human, and maintaining it in a way so that the all of the extracellular components of the tumor that make it a human tumor, and that also tend to trip up a drug and challenge a drug and create the difficulty of whether a drug will work or not in a person. All of those characteristics are maintained in the mouse so that you're not getting an easier version of a human tumor. You're getting the same difficult version of a tumor in that mouse that you would in a human. So that's why it's highly predictive. But of course, it's only a mouse, so the ethical issues aren't the same in terms of the experimentation. Now, what else does de-risking do? Well, we're not here to tell you what the future value of this company will be or what it will do to any future investments. That's certainly not our role or job. But just to illustrate conceptually what de-risking can do to the perceived and assessed valuation of a company when it's successfully done. We use a model that's widely used and respected, a risk-adjusted net present value model by a venture type firm out of Seattle that's used to model new biotechnology based on factors such as future revenue, cost, and time needed to get it to market, and the risk of failure. Now, the last part, the risk of failure, is what we're going to, that's what we're going to drive down with the data at the next phase. And when you can drive the risk of failure down, the valuation can rise dramatically, so that the ability to obtain capital and certain valuation and to secure the sort of partnerships you need can be dramatically influenced and improved based on data results that show the probability of this drug succeeding eventually is higher than it was at a prior stage. And that's the rationale behind what we're pursuing next, the next phase. So, just an example, these aren't, necessary statements or projections about anything about us in terms of what our future valuation. These are illustrations, but making certain assumptions and plugging in certain variables in the model, you see how going from a 5% risk of probability of success, that's very low, and that's a very small number that we're saying, only 5% chance of success, to a 40%, even just up to 40% chance of success, can have a dramatic impact on the valuation of a biotech. So we've had past and current relationships and potentially future relationships that have been very useful, instructive, and potentially promising for development relationships and possible partnerships. Although we want to maintain a low profile, our research has resulted in some invitations and selections for partnering meetings, conference presentations, etc., with some of the world's foremost organizations. And these were these were basically all opportunities to determine what the level of interest is, both now and potentially in the future. And we received very promising feedback from many of these entities. I'm not going to specify or talk about the details of any particular discussion, but the common denominator in our discussions was that they would like to see in vivo data, the type of data that we would more than obtain in a PDX trial involving DBX31 at the next stage. And this kind of data could have a significant impact on the possibility of future partnerships with these or possibly other entities. So basically our short-term goals and game plan is conducting PDX tests. That is the in vivo research using these highly predictive patient-derived xenograft platform is the first goal. That's the most critical one. If we have enough capital and it's available to us to perform the same sequence of work, we would then also conduct a phase 1 vet trial. That would help us to potentially achieve the third component of the strategy, next phase is the third phase of strategy, which is establishing partnering or licensing agreements with possible co-development or licensing entities, or also in-house, maintaining in-house, and just proceeding forward with the self-development strategy. And we're not saying that we're committing to a particular strategy at this point. We're saying we're going to let the data that we obtain next phase provide our options for us and pursue the ones that make the most sense for us and our investors. And basically, at the most recent and current stage, our core team is reflected up here. But the key point I want to emphasize is that while strategic and operational decisions for the present research are going through the current core team, primarily, of course, in-house within DBS, myself as the CEO, this is a multidisciplinary program that requires complementary subject matter expertise, ranging from protein engineering to regulatory compliance to clinical trial execution. So there's no single person or group of people right now who possess all the skill sets we need to drive this all the way to the finish line. We're going to have to bring in specialized expertise that we don't have on board now and that we can't afford right now. But the key to doing that, again, is getting the de-risking data at the next phase that will allow us to get the capital, financing, and the credibility and relationships that will allow us to basically expand our team or to move members of the team in and out to achieve the purposes necessary for that particular phase development. So there's no one expert who knows everything about everything to get this molecule from here to there. It's a set of experts, all of whom contribute at different times and then move out of the way or collaborate with other experts who then come in. But so even though we've used these individuals today and they've have tremendous value to us, and by the way, the names have been changed, just to let you know, it's that listed down here in the disclaimer language. The names are changed because they're getting a lot of exposure here. And frankly, we've been contacted by, I think, some suspicious parties from various sources. They possibly corporate espionage related, possibly related to just international hackers and things like that. So basically for their protection, I'm changing their name here, but their credentials, their titles and so forth are all absolutely the same and unchanged. And of course, myself at the top here, just in terms of background, you know, there it is. I have Bachelor of Science and Doctor of Law from University of Wisconsin-Madison. I obtained my Master's in Industrial Management from Cornell. I have, like I said, experience as a former operations director at Lucent Technologies, Bell Labs. And I've had a long time interest in biotechnology fields, bioscience fields. I started out when I was in AP Bio in high school in Chicago. My teacher, who was at the University of Chicago, a biologist, she introduced us to particularly cell biology and a lot of opportunity to get involved in the whole field at University of Chicago. And I just took it with me through college. Started out to consider becoming a pre-med, didn't complete that. Instead, you know, took all my pre-med requirements, but instead decided, you know, I think you have to be a little bit smarter than I am in order to have people's lives in your hands. So I said, I'm going to leave that medical school thing to the real profession. But anyway, you know, I did proceed down a different path, still maintain that expertise, and got an opportunity to apply it in my professional life unexpectedly once I got to cut my teeth in a Fortune 500 company at Lucent Technologies, Bell Labs. So anyway, you know, that's pretty much the you know, nuts and bolts of that. I apologize for that technical problem, and you know, we're at the end, and hopefully things will last long enough for me to finish out the rest of this.
Wonderful. John, that was absolutely wonderful. You can stop sharing your screen, and
Okay.
then I will go to the Q&A. We've already got quite a few in here. Guys, if you have any additional questions, please don't hesitate to put them in the Q&A so that John can answer them this evening. So Horace says, what happens to the debris after the kill? So in that one, I feel like it might have been test 3 of 8 or something, but you showed the picture of the solid mass, and then you see the second picture, it was dispersing some, and then the third, it was dispersing even more. I think the question is, with that debris, what happens to it? And there's actually multiple questions in here about the same thing. Sharnetta says, what happens to the cell debris after it explodes? Is it absorbed back into the cell, or does it disintegrate?
Yeah. Well, apoptosis is a naturally occurring process as well. Apoptosis happens in normal biological and metabolic processes in the body. It just doesn't happen on command and at the level that we would try to make it happen to eliminate a solid tumor mass. It doesn't occur. Your body doesn't kill that amount of cells at that time that vigorously and aggressively and on command. So the body has a normal process for excreting and handling byproducts of apoptosis. Probably a combination of both absorption of some components can be reused, or going to be basically can be metabolized or reused in various, but much of it probably just basically excreted as waste matter through our various excretion processes, the way we excrete various types of waste in other ways, naturally.
And so what you showed us was at the 72-hour mark, but am I correct to assume that the treatment for this type of drug would extend beyond that? So a patient would be taking this for a longer period of time. You were just isolating the progress in a short period of time, right?
Correct. Yeah, that's right.
Continue to break down. Is that a true assessment?
Yes, I mean, yes, that's the ideal and the mechanics of it is that if you have, let's primarily talk about a solid tumor mass. We don't know about hematological malignancies, that like leukemia or myeloma. We think that there's possibilities of application here, but we haven't tested yet. But just there's enough solid tumor mass cancer out there to deal with that's great. Assuming this is like a mass, and it operates, it would interact with cells that it has physical contact with, that it can actually touch. So if it's floating by and bumping on the outside, it would then cause those cells to go, and then they would shed, and imagine pieces of them would shed off. That means the cells underneath those that were not exposed would then be exposed theoretically, and those would presumably be bound, and then those would then break off, and those would shed. So imagine like a peeling out of an onion, where each subsequent layer gets exposed now to the molecule they weren't exposed to, and that would theoretically be how it works. So eventually, the objective is, let me just emphasize, even though we got great results and great data, we're not saying that we have all the answers at this point. I can tell you how this would, or even if it would work clinically yet. But we have, there are examples that exist pharmacologically and biologically in the world that we believe that operate in a similar kind of fashion, but maybe in a different disease or different context that we can infer would also apply here and probably result in the same manner and same method and process.
Okay, that's a very good point. That part of what you're raising the money for is to be able to continue the testing, right? And possibly test it on animals and then, eventually on humans, but we're not there yet. Right. Okay, I just want to make that clear. So Horace says, I'm gonna actually read Sharnetta. Sharnetta and Horace are on the same page this evening with their questions. So Sharnetta's question says, has there been any cases of the cancer returning after the cell has achieved cell death? If so, how long after the treatment did the cancer return? And then Horace's question is, does the patient still die after the 90% improvement?
Well, let me apologize for creating that impression that we're dealing with a human patient yet, and that we've obtained answers, that kind of clinical proof of how it behaves and how it operates. We don't have that yet. We can make some, I think, reliable reasonably reliable projections and estimations of how things would probably work or could most likely would work. but it would be, scientists tend to be very conservative in their statements, at least credible scientists are that way. And you should be conservative in making projections about what would happen in the future and how it would work. And that's what I'm not trying to be evasive, but that's what I'm going to do, simply because we can't state something factually that hasn't happened yet. What I suspect or believe would be the situation, I think what you're kind of asking is, maybe, even though these were in vitro, these were in non-animal and non-human context, right? You really should still, if you want to call it flask or test tube kinds of, let's think of that context. In terms of the cell coming back, the cell, once thing about apoptosis is that cell's dead permanently. It's never coming back. Once you do induce apoptosis, those cells that are apoptotic are dead.
They're not going to reform. And when you did your test, you didn't see them reform in the petri dish. Once they obliterate, they obliterate.
Oh no, they're gone. Yeah, there's no way. Yeah, there's no way. The fundamentals of my lamp over here is now around me. I didn't see that. But the fundamentals of cellular biology make it impossible for that cell to reform. It doesn't synthesize and come back to reintegrate after it disintegrates. There's no way it can happen. Okay. Other cells, it's possible that DBX31 would not kill all the cells in a particular tumor, right? And also, it's possible there might be cells that have properties that, like the SKB03, are mutated in a way unlike the other cells that makes it resistant to even DBX31. That's possible. I'm saying because we haven't encountered that yet. Nothing we've found resists it, but it's possible. And frankly, the percentage, the lower level of percentage, less than 100% rates that we got, could be due to, there could be patches of cells that are resistant, are mutated in a way that makes them resistant to DBX31. That's always a problem. In fact, that's basically how most cancers work, advanced stage cancers work, or stage 4 cancers. Even when they have positive initial treatment results, that's the way they work, is that when they say the cancer is responding and the cancer has reduced and eliminated and gone away, for stage 4, usually hasn't gone away completely. It's still, there are microsets of those cancers still around, and they're usually what's called stem cells, or they're like their most powerful resistant cells that are left. You just can't, they don't show up on a imaging, so they look like they're gone, but they're still there at a very microscopic level, as you can see. And those can essentially reproduce and reform again. And that's why usually when you hear about someone who had advanced stage cancer and they got a treatment, it looks like they said, I'm better now, and they're good. And they said, I'm good to go. And then you hear about many years later, their cancer comes back, or they were doing a cancer set, and this time there's no treatment option, and they die from that. That's because that cancer is now composed of those cells that were resistant, and I use the term immune. That's not correct, but it's a common term we all use. I'll just use it as a layperson, but resistant to or immune to those therapies. But they were so reduced in numbers to such a small number that they were basically harmless to the body. It wasn't a big tumor again. It was a very small one. So it was basically harmless in a person lived from. But once it produces and reemerge as a large tumor composed of just this super type of cell that's now reproducing cancer, and that tumor mass is not responsive and treatable anymore. That's possible with DBX31. The good thing about DBX31 is, even in that case, it demonstrated such a high rate of reduction and cutting into the makeup of the tumors that all did respond to it, that even then it's highly likely when you have that kind of apoptotic level that you can probably reduce the tumor burden, is what it's called, and quite plausibly extend that person's life quite a while before they would succumb to it, or perhaps permanently before they succumb. They would just have some cancer in their body, but it wouldn't ever rise to a level that it became damaging to the organs and cause, death.
Thank you. Horace says, what prompted you to take this particular angle in your research?
Yeah, we weren't conducting research looking for a cancer therapy. That wasn't how we were actually led into this research by evidence and data, observational reports of observational data that there was a naturally occurring compound that had a folk medicine effect, if you will, that actually had real world results. And it came from a credible source to me. It came from connection through my my family tree, my family legacy. And it was brought to me because they trusted and respected, I don't know why, aspects of my life, and said, well, you ought to know what to do here with this information. What do you think? Is it worth pursuing? And I said, well, yeah, this is how you would pursue it. And you'd form a company, and you would then conduct research, and you would then evaluate it and have it tested by a third party, blah, blah. And so I said, that's the way to do it. And so this was coincidental, and what they call it serendipity. It was so what gives us the most, I guess, confidence about this is that I was skeptical, as would be most people, when someone comes to you and tells you that. It's not that you think, I don't think they're lying or anything. I just think, well, was it perceived correctly? Was what you thought was there, was that really what actually happened? But I said, okay, we'll take this data and we'll test it in a controlled environment, and we'll see if it reproduces the results. And to my amazement and delight, the data so far has reproduced things that I couldn't have independently created. I'm not smart enough, and frankly, I'll say this: I don't think anyone's smart enough to be able to simply decide this is what a breakthrough in cancer is going to come from, or where it's going to come from. You can't. There's a degree of luck and coincidence involved in almost all great scientific breakthroughs. Whether the scientists admit it or not, almost everybody benefits from some observation that they didn't entirely plan or expect. So, and that was the case here. We got information. We then hypothesized about the information, what it would take to evaluate it, and what it would look like. And it turned out to, so far, we've been rewarded. I'm sorry. I think you're on mute, Tawan.
I am. I'm sorry. How long have you guys been working on DBX31?
Since it was actually introduced and brought to my attention for a long time, since the early 2000s, that it was brought to my attention, and I said, well, this is going to take a good amount of just research. Both field research and then literary research. And I have to pound the books and go through the medical libraries and the science labs and do a lot of comparisons and evaluation of, right, what is this compound like compared to this, and what are the organism source that was from, to get a little bit of zoology and background together in terms of, right, let me learn as much as I can about this organism and its and the various properties. So it took a while just simply to even come up with a coherent, compelling thesis to take to a credible institution like Cornell, for example, and get their respect and get their attention. And that was in the later half. That was closer later toward that data from Cornell really came together about 9 years after that. So, okay. Yeah.
Okay. Kendall says, what's the timeline for the short-term goals for the preclinical research?
Sure. That would be about those short-term goals, timeline of the first part, the PDX test component, which is the most important. That's about 12 months. That's about 12 to maybe 14 months. About 12 for it. So we're looking at about we have to recultivate, conduct some additional cultures, tissue cultures, then do some additional amino acid sequencing. Then we have to do we have to basically get it synthesized. That's an important part, is get it artificially synthesized by a CMO. So we're going to reproduce it through what's called recombinant DNA technology. And all that stuff would take about, like I said, about 8 to maybe 10 months. And then about the last 2 months would be about the time for the actual studies. So, I mean, you got to factor in, oh, hey, what happened tonight? You got to factor in stuff that you don't foresee, so probably throwing a couple of more months on top of that. So like I said, between 12 and 14 months, I think, to get that PDX results. But the other thing I do want to share now in relation is that if we don't get the full, if we don't get a sufficient capital raised to do that at this particular campaign, a couple of options. One, well, we're going to move forward with it, just proceed forward with it, with the money that we do have, and simultaneously while that's happening, I can initiate another capital raise, except this time probably a private placement. So probably instead of doing that, private placement. The reason why, because we've been contacted by a lot of, if you will, accredited investor types that are saying they would be interested in a private placement, and we have connections, but not, what their thing is, maybe just not angel investing. They're whatever snobs or whatever. I'm just saying. But that's something we could pursue after this. And we have there are quite a few interested parties that we would decide. If we didn't do that.
What's the total amount you're looking to raise? I know you're at the $83,000 mark now. What's your total raise desire?
We would like to get to do the PDX mouse test. We would like to, we'd like to get in the 150. We'd like to do 150,000. I mean, maybe minimum like 140, 130. I will make up that difference like I have in the past. But we'd like to end this with that much, at the end. Is that going to happen? We don't know, but can we move forward? If we move forward, what we have now, there's a more economical approach that we could take, and we would possibly take alternatively. That is using PDX models, using the same PDX model, patient-derived xenograft models, except not in mice. Instead, it would be using what's called zebrafish models, and they are less expensive than mice. They're highly predictive. They're not quite as predictive as the mouse models are. Those are the gold standard. That's the absolute indisputable top of the line, highest standard. But they cost more, and they require more compound for us to test them in those. So we have to generate and pay for generating more DBX31, if you will. The zebrafish give a high degree of reliability, and they're used by the top contract research organization that does these type of tests in the world, at least one of the top, Charles River Labs. And so Charles River does use them. They have a good, very reliable reputation. They wouldn't deal with garbage. So it may not be a gold standard like the mouse PDX models would be, but I think maybe closer to maybe like a bronze standard, or a silver standard. So it might not be a gold medal, but it's still a pretty high level, standard. It still would be like getting a medal on, it might not be the number one, but it's in close category. But it would cost a lot less, and on both the actual tests themselves and the amount of compound that we have to manufacture in order to do those tests. And so it could definitely be, if nothing else, a precursor. Even if we didn't get that full raise amount, we could move forward with studies in that model instead of the mouse models, and then get the data there, and then maybe do the mouse models after that at the next phase, which then gives us the gold standard, as well as some other models. Basically, it would give us more data, that we need right now in animal models, but at a lower cost.
Gotcha. So so hypothetically, if you got all of the capital you needed, right? So that money was no longer an issue, right? What's the time frame that you believe you could get to completion? So we're taking the barrier of capital completely out of it. You're fully funded. What's the time frame to FDA approval, or at least FDA submission
Okay, great. All right, so we assume that, and we also assume that we get we get the data, continue to
Yes.
replicate the kind of results that we've seen so far. No massive surprises happen. It looks good. All right. Well, we project, I mean, we can't speak for the FDA, of course, but I think we can confidently say that we might be looking at something in the order of 3.5 to 4 years for getting early, what we consider a preliminary approval or an accelerated approval. And that's still FDA approval. What that means is that we would get approval short of a phase 3 clinical, fully completed phase 3 clinical trial. We would then be allowed to, in other words, based on earlier stage data, we could get a temporary or interim approval by the FDA, because we would be addressing an area of medicine that has a high need and that it's unmet right now, and that people are basically dying literally for lack of. And that's an area that the FDA would be willing. It's very similar to what happened with COVID here, right? And normally what happened in terms of getting a vaccine out, it doesn't happen like this. I can tell you that you don't get this fast. People may think it took a long time, but under normal circumstances, no, this is an insanely short time frame. And that's what the FDA will do under these life and death circumstances. The FDA doesn't want to be accused of killing people, blocking people from access to it. So there are, I think, a number of cancer types that provide us with options for getting accelerated early approval that, we potentially have the ability to show benefit in. So that gives us, when people might hear this, say, well, that sounds he doesn't know what he's talking about. That's insanely prematurely. Yeah, it's a normal slow path for a drug that's being targeted for a primary stage cancer, for which there are already therapies in place, and this just might be a different one or a slight improvement. That's true. But when you're addressing a disease where it's a high level of death and morbidity, and there are no current options that are viable, the standards, they relax the standards dramatically, because the safety question becomes more secondary at that point, because it's like, gee, what could be more unsafe than advanced stage cancer, So that's where you have, and that allows you to get that approval to use it, to sell it, while you work on getting that other data in the background simultaneously.
Right. Okay. So Horace has a couple of questions. I'm going to combine two of them. So he says, where did your family get the original data? And then, was the original substance from the family an herb or drug?
Wasn't an herb. Well, I can't say exactly where we got the original data, other than to simply say there's no problem with ownership of this. Okay. There's no problems in terms of nothing's been stolen. It hasn't been pirated. It wasn't. There's none of these issues involving biopiracy that some companies are accused of in the Amazon forest, taking things from indigenous tribes and people. There's none of that here. In terms of what the classification of it is, no, it's not an herb. And I understand what he's talking about. I think from a legal and from a commercial perspective, and from a patent protection perspective, it's not an herb. It is a bio. Well, it's a, think in terms of think protein, that kind of class of drug. Class of molecule. Think that, and it's not herbal. So, and also eventually, from a commercial and legal perspective, you'd want to synthesize the drug and basically make an artificial form of it anyway, just because, one, it's quicker, cheaper, easier, faster, and probably more stable to make it that way, but it also then gives you legal protection in terms of intellectual property rights that you wouldn't get if you used the natural original molecule all the way through your commercialization.
And does DBS already have the patent on DBX 31?
It's considered patent pending.
Okay.
Provisional patent stage. You know.
Wonderful.
Yeah, and it's, you know, there are pros and cons to that, but I think the pros at this point outweigh the cons, because let's say if we did have final patent completed stage, at this point the clock would be going tick tock, tick tock, tick tock. You're burning off your patent term, and that's never good from it. You're losing patent protection years of. Even though you do get patent protection when you get an FDA drug, you don't get patent protection. You get FDA approval protection when you get a new FDA authorization. So you still get us over a decade, like about 12 years of protection on an FDA anyway. But so even if you don't have a patent, you can still get protection from competition with the FDA. But that's not ideal. You still want that, to me, that full patent term for just a bunch of different reasons.
Right.
Yeah.
And then Horace's final question is, what is the estimated time frame on the de-risking?
Yeah. Well, okay. Yeah. So the immediate de-risking goal. Excuse me one second. I'm actually having a little tickle in my throat.
Okay. I'm going to take a minute to take a sip as well.
Okay, great. Yeah. And by the way, thank you for the questions, Horace. The de-risking time frame, would be the initial de-risking time frame would be the completion of the PDX models, the trials, the PDX, patient-derived xenograft trials. So again, that would be assuming, depending on how much we get and what type of PDX models we can conduct, that would probably be in that 12 to 14 month time frame. If it's the non-mouse PDX, if it's the zebrafish PDX model, then that would probably be just about the same time frame, maybe a little bit shorter than that. Actually, those it's easier again and quicker to do those. So might be a couple of months sooner than that. In terms of final de-risking, you have to, that would be basically an FDA approval, and that's the ultimate de-risking. That's the final de-risking, and some clinical trial phase. That's the only thing that finally de-risks everything, and that would be, like I said, that probably that 3 to 4 year time frame. But you can have a high degree of confidence of de-risking, and like I said, and that by the time you got that PDX model done in 1 year.
Okay. And then the final question is Horace, and Horace says, the concern was that drugs typically have side effects, whereas more natural types of applications have significant side effects. Are you going to continue to monitor the difference between the more natural formulation and the synthesized form?
Okay. Well, I'll short answer that with, I could kind of just some other, I was going to maybe address and respond to a certain aspect and a certain assumption in there, but I just, in the spirit of the question, Horace, the answer is yes. Yeah, we would certainly evaluate basically not just even one synthetic form, because there are theoretically multiple forms, molecular forms we can create here, and I'm not getting into that now, but that's also part of the possibilities for de-risking and figuring out ways to maximize the probability of future success, is that there are multiple ways to basically skin a cat here. Well, bad, I'm sorry, especially when we're talking about veterinary products, that's a bad metaphor, but there are multiple ways to basically get the job done. I think when it comes to possibly creating a molecule that triggers that death receptor. It's called molecular agonist is the term. Basically, that just means a molecule that looks like another molecule, but isn't exactly a molecule, but kind of binds to the same receptor that one does. And the different ways to do that. So, we will look at actually multiple forms of DBX31 and its agonists. That means sort of, if you will, similar molecules that can do the same job.
Very good.
Yeah.
Any additional questions? This was such a great, I always say the best part for me about the Bet on Black Pitches is how much I learn about a new industry at the end of the evening. And you did not disappoint, John. This has been very educational. Any other questions for John before we give our final closing comments? And Horace says, thank you.
Thank you, Horace. Thank you. If it weren't for you, I'd be sitting here probably looking at the wall for
I love this community. We always have someone in the audience that like knows enough about the industry to keep the conversation going. I love this community for that.
Absolutely.
Awesome.
Absolutely. Very, very much.
I'll step in and give my final comments and then turn it over to John to close us out. First of all, to John, thank you so much for coming to 10K, but more importantly, for dedicating yourself to this project for so long, right? And trying to come up with a solution that's going to eventually help so many people. So thank you to you and your team as well, because as you stated, it's not one person coming up with this. It's the input from a lot of different people. So thank you to all of the people that have assisted you with this and that will continue to assist you on the journey. I'll say this to the community, as I always say, we're not, Cherie and I are not financial advisors. We don't play one on TV, right? So we're very open and honest about the fact that we are not financial advisors, but what we are two entrepreneurs that understood there was a need in this space for our people to learn about it, both from an entrepreneur standpoint, right? Entrepreneurs needed to know this was a way to raise capital, and then investors needed to know that this was a way to earn potential wealth. And so we created the 10K to help you on that journey. I will tell you, equity crowdfunding is not going anywhere. Talisha actually sent us over a graphic this week, and $500 million was raised in 2021 via equity crowdfunding across all of the equity crowdfunding portals. So that's, the WeFunders, the net capitals, as we saw this evening, the start engines, all of those. $500 million has been raised for founders. So the space is not going anywhere. It's just starting, right? And we hope to grow 10K and be at the forefront of it, helping our Black founders, because that's our focus, helping our Black founders get the money that they need. So if you haven't already shared 10K with your family and friends, please do. We know that the more members that we have, the more people that we can get looking at these campaigns so that they can help the founders raise the capital that they need. So please, if you haven't shared 10K, please share it on your social outlooks. I always tell you to share the campaign post, and I want you to do that definitely, even more than sharing 10K. I want you to share theirs, but I also want you to share us as well. Also, if you are interested, we all have these resolutions that we're making, and if financial wealth building is on your list, and if you're here, it probably is, and you want to talk to a financial planner, please consider upgrading to our platinum package. We're going to have some testimonials here soon from the people who are finishing up their 6 months of the financial planning sessions. But please consider that. It would just be for all of you, because you're all members, an additional $247. So it's that $100 you've already paid applies to it. With that said, in regards to DeoBioSciences, please go on the web, the link that we've been posting. Please go there, read through all of that information. John and his team put together some great information for us. We saw the presentation, but there's so much more out there. Some of the financial numbers are out there. Please take some time and look at that. We call that doing your due diligence. Then make a determination on if this fits in your portfolio. One of the things I know to be true is I've never had something this cutting edge and at such an early stage presented to me as a potential investment opportunity. So, to John and to founders like him, I just want us to thank them for taking a risk in equity crowdfunding space, because it's a lot of work, right? And it's not always successful for some of them, but it's a lot of work. It's always a lot of work. That part doesn't change. It's always a lot of work, even though it doesn't always render the reward. And so I just thank all of the founders, not just the ones that have come here to 10K, but all of them for taking a chance in the equity crowdfunding space, because it's because of them we have these opportunities, right? For those of us who are not accredited and don't have, the end to being able to get into an IPO and all of those things, I say thank you to those founders who have, taken the opportunity of equity crowdfunding and turned it into an opportunity for us. So John, with that, I'm going to ask the family to share your campaign, share, right? Please share his link. We ask that you copy the link right now that we've been putting in the chat. If you go down to the bottom of the chat, you'll see next to the smiley face, 3 little dots. If you click on that, you'll be able to see how to copy the chat. And then that way you'll have that link, or you can just take that link right now, copy and paste it somewhere else so that you can easily share it. We'd like for you to use that link, because that's the link that we'll be able to come back to John and say, hey, John, we sent, 700 people to your campaign, So if what's important though is that you just go to the campaign page, whatever link you use. But if you use ours, we'll be able to report back to John some stats so that he can know if this was a good return on his investment in 10K. So with that, I want to thank you all for joining. John, I'm going to turn it over to you for final words, sir.
Okay, great. Yeah, I'm not going to start with some great dramatic wrap up, even though I thought about that for a while. Then I said, well, you know what? I want to give them some practical, and I've been excited, but I think it's very important to consider this. This is for not just for us, but for any investment you consider in equity crowdfunding. It's very important from your perspective as an investor to think about the exit strategy. What's the exit strategy? That's really that's the only way you can kind of get a return on your investment, as you have to find what are the options for you investing in. I mean, if it's, I think the 3 components, the 3 typical aspects of exit strategy you have to be very mindful of are, is this a potential IPO candidate? All right. Is it a potential, alternatively, M&A, mergers and acquisition candidate? All right. Or is it potentially none of those, or maybe all of them? You're going to have that too, and a long-term hold with high net profit potential that will result in dividend distributions. And this is not investment advice. I'm not telling you invest in a specific. Well, obviously I have a personal interest in my own, but this observation applies across the board to any investment, not just even in equity crowdfunding, period. This is just this is bread and butter advice here that you have to consider. And no matter how great or sexy something sounds, if it can't provide you with a way to get your money investment out, or you get a stream of, or to sell your investment in it, then it's kind of bound. You got to be prepared to basically give up that, let that investment almost go, because you have to be able to extract it somehow. So I would say, in terms of net capital and DeoBioSciences, one important factor you need to think about is that they provide what's called a secondary trading market, and so that's basically it's a stock market. It's not the major public exchange like the NYSE, New York Stock Exchange, or NASDAQ, but they do provide to their investors an option. It's called Rialto Markets, and it's an alternative trading system where basically, if there's a whole, you have to learn this, educate yourself about equity crowdfunding, but specifics. But in general, there's a hold period by the SEC in equity crowdfunding, meaning you can't sell your investment for about approximately a year. Right? After that, if you can find, if your company is listed on a market and it provides access to a secondary trading market, then theoretically, at least, you have the potential to sell your whatever those securities are, right? And that's something you should be very considerate of. Can I sell this later? So that, hey, maybe within a year, as little as a year, I might not want to ride this wave for the entire 4 or 5 years or 20 years or whatever. Maybe I say, you know what? I'm good with, wow, there's a great event that happened in a year. I'm good with selling it for whatever the market is willing to pay for it at that time. That's what we consider. If it's not, you know, that doesn't exist as an option at trading market, which, like I said, it does at Net Capital. That's one. Then also consider, is this company potentially something that could be acquired by someone else for a merger? And that's one thing you have to think about relative to us. Make that decision yourself, but check out. I'm sorry. Oh, Tawana. Oh man. Okay. I don't know if Tawana's if we got cut off or something here. It seems like Tawana's frozen here, and if you can still hear me, folks, I don't know. I'm going to. Looks like we're still connected. At least I still see some. Maybe not. Let me look and see.
Yes, so we're still connected. You can continue on.
Okay, great. Thanks for telling me that.
You're more than
I'm going to hold on till Tawana. There, great. All right, Tawana. Glad to have you back. Boy, you missed everything. You missed the last part. That was the best part. All right, everybody. Well, hey, it was great again being with you tonight. I'm glad you got those last 25 seconds. That was worth everything.
Thank you so much, John, to the 10K community. Again, Happy New Year. You know, we've got a full calendar for January. I've got February almost wrapped up. And so we're booking ahead to try to impress you. Next week's founder is actually a return founder. He wanted to come back because he felt like the 10K community was such a good community, and he had some success, so he wanted to come back in the new year. But then everyone else for the month of January and February are brand new. So super excited to share the calendar with you guys. I'll be working on March this week as well. And we're going to keep doing this. You keep coming. My promise is to keep doing it. So with that said, have a good evening. Don't forget, capture the link before. I'm going to give you a minute if you haven't already copied the link. I'm going to give you a couple of seconds to do that. If you want to copy the chat, remember the 3 buttons down at the bottom so you can save the chat, but grab that link. And have a wonderful evening, and we'll see you next week, guys. Thank you again, John.
Thank you, everybody.
Bye.
Bye-bye.